Right, good evening, people. Yes, so today we will discuss BioEve paper 13. BioEve in the paper. Lamai, can you find the paper? BioEve 13. Right, BioEve 13. Yes, let's go chat. Papers there. Paper, we see high paper. Telegram. Right, people. Um, take that BioEve 13 paper. We'll discuss the MCQs as usual. Can you hear me clearly now? Did you answer BioEve paper? Did you receive BioEve? Right. Right. Yes. All right. Now, people, I will tell you the BioEve papers and the discussion. We are uploading this to the website, and so you can go to the free videos and then you can watch it. Right. It's present under free video. Log into Advanced Online website. Go to free videos section, and you can see bio free videos. You have BioEve, chemistry free videos, chemistry Eve, right? Physics free videos, physics Eve. So if you miss any paper, you can get the video. You can watch the video, right? First question: the number of essential elements found in a human body. Number of essential elements found in human body. In yeah, number of essential elements found in human body. Out of the elements of the living matter is, out of the elements of the living matter is, um, how many human body you going to take, people? Essential elements found in a human body out of the elements of living matter. Um, we said plant 17 and then human it's 25. Ne? So I think I can go with this one as the answer, 25. So I think you have taken the right answer. Question number 2, nitrogen containing carbohydrate is hemicellulose, inulin, glycogen, chitin. What is your answer, people? You have to give me on the chat line the answers now. I'm taking, people, fourth one, chitin as the answer, chitin. And then next one, what is the molecular weight of cellulose molecule? What is the molecular weight of cellulose molecule made up of 500 glucose molecules, 500 glucose units, right? What is the answer? What is the answer that you have obtained for that? Right. Now, what is the molecular weight of one glucose molecule? What is the molecular weight of one glucose molecule? Then you have to work it out for 500. You have to work it out for 500, right? What is the 180? 180 into 500. But then the question is not over with that, right? Why it is not over? You have to deduct from this value, right? 500. How many glycosidic bonds formed? 499, right? So here we can see that 499 glycosidic bonds present. So in each glycosidic bond, how many water molecules? One water molecule goes. What is the molecular weight of water? Molecular weight of water is yeah, 18, right? 18. So then what is the answer once you right work it out? What is the answer? Answer? Which one? What is your answer? Come on. Come on, come on, come on. Yeah. What's the number? What is the value? 81,018. 81,018. Is it? I got third one. I got it as third. Can you work it out once again? Fifth one. Yeah, fifth one. Yeah, I think I had to then calculate that part once again. Uh, let me check. Did we go anything wrong? No, ne? Okay, ne? 18 into 499, 8982, ne? 8982. Then 180 into 500, 90,000. 90,000. So 90,000 minus 8982. Yeah, you are correct. Right, you are correct. So answer has to be fifth one. Eh? Answer has to be fifth one. So we'll take the answer as 5. Answer is 5. Answer is 5. Answer is that. Next one, people. Next one. Question number final glucose units. Next one. Which of the following is not a function of monosaccharide? Which of the following is not a function of monosaccharide acting as energy source? Acting as energy source. And then not a function, eh? Not a function. What's your answer for that question, people? Question number 4. What's your answer? What is your answer? Come on. Yes, fifth one. Some people say fifth one. Act as a storage component of the cell. That is correct. So we'll take fifth one as the answer. Monosaccharide storage material, wrong, right? Question number 5. Question 5. Select the correct statement regarding nucleic acid. Select the correct statement regarding nucleic acid. Right? Major component of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Component nucleic acid, potassium, phosphorus. Wrong, neither. First one is wrong. And then consist of branch long chains. No, and they are not branched. And then next one, people, phosphate group gives basic properties. No, acidic, right? Wrong. And then contain carbohydrates. That is correct. It has carbohydrates. And then act as a structural as well as storage component. Structural and storage, wrong. So we have the answer, people. Contain carbohydrate. We'll go with the fourth one as the answer. Fourth one. Question number 5. Question number 6. Select the incorrect statement regarding light microscope. Incorrect regarding light microscope. What is your answer, my dear students? The maximum magnification 1000 times. Maximum magnification 1000 times. And then maximum resolution power is 500 times as a resolution power of the human eye. 500 times as a resolution power of human eye. That we had to work out, neither. And then people, cellular organelles cannot be observed. Cellular organelles cannot be observed through it. Wrong. We can see some cellular organelles. Right. And then living and non-living specimens can be observed. Living and non-living specimens, yes, that's correct, people. Resolution power is limited by the wavelength of the light. That's also true, neither. So we have the answer, people. I'm taking third one as the answer. Do you agree with me? I don't think there is a big issue. 500 times, you can work it out and then see, people, how this 500 times comes. How 500 times comes? How do you find that? It says, people, maximum resolution power is 500 times as the resolution power of the human eye. What is the value? 0.1 millimeter. Light microscope, 0.2 micrometers. That is equal millimeter into micrometer, 100 micrometers, 0.2 micrometers. So that means, people, 1000 divided by 2, neither. So that's 500. That we get, nah? That's correct. 5 times is yes. Correct. Right, next one. Question number 7. Question number 7. Question number 7, select the incorrect statement regarding cell wall of plants. Incorrect statement regarding cell wall of plants. Primary cell wall is always permeable. Primary cell wall is always permeable. That is correct. Secondary cell wall lies outer to the primary cell wall. Secondary cell wall lies outer to the primary cell wall. Outer to the primary cell wall. Secondary cell wall. No. Outer to the primary, no. Inner to the primary. Neither. So second is wrong. It is an extracellular non-living structure secreted by plant cell itself. Extracellular non-living structure, correct. It controls the growth of the cell. Yeah, correct. And then people, cytoplasmic strands run through the pores of the cell wall adjoining cell. That correct, neither. What do you call these cytoplasmic threads, people? Plasmodesmata. So my answer is second one. I think people, you can agree with me without any issue. Ne? You can agree with that second answer. So we will question number 9. Can you check question number 9? Question number 9. Question number 9. Question number 8. Sorry, question number 8. Question number 8. During denaturation of protein, primary structure is destructed. During denaturation, primary structure is not damaged. Wrong. Three-dimensional structure of the protein molecule is destructed. Correct. Peptide bonds are broken down. Wrong. Change in the sequence of the amino acid. No. Reduction of amino acid number happens. No. So therefore, my answer for this question, I'm taking second one as the answer. Right? Three-dimensional structure of the protein molecule is destructed. That is correct. All the other statements are wrong. Question number 9. Which of the following combination include lipids only? Which of the following combination include lipids only? What is your answer, people? Can you show the calculations for the sixth one? Showed me. You multiply and then find, people, how many water molecules. Come on. Question number 9. Which of the following combination include lipids only? Which of the following combination include only lipids only? Right? What is the option? Chitin, phospholipid, steroid. Chitin, not a lipid. Ne? Wrong. Then second one, people, wax, terpene, actin. Actin is a protein. Ne? No, wrong. And then suberin, vitamin D, and cutin. Correct. Third one is okay. So I'm taking third one. I think you agree with me. Some of you have sent me the answer as 3. So I do agree with you. Third one is the answer, people. Question number 10. The other two statements are also wrong. Collagen, you can't take the fourth one. Pectin is a carbohydrate. No, you can't take that. Question number 10. Question number 10. Which of the following is a complementary event in meiosis? So what is retinol? Retinol is vitamin A. Can you write there? Vitamin A. Vitamin A. What do you know about vitamin A? Water soluble or insoluble? I didn't see that. Retinol. What are you going to write for retinol? Vitamin A. If it is vitamin A, is it water soluble or insoluble? You know this. You learn it. Water soluble or insoluble? Insoluble in water. Can write there instead of retinol, cut that and write there vitamin A. You know vitamin A. Huh? Right. Question number 10. Which are the following compulsory event in meiosis? Compulsory event in meiosis. Pairing of homologous chromosomes. Pairing of homologous chromosomes. Crossing over between chromatids of homologous chromosome. Formation of chiasmata. Formation of 4 identical daughter cells. And then duplication of centrosomes. What is your answer for question number 10? Can you tell me the answer? Right? Which of the following is compulsory event in meiosis? Meiosis. You are coming with different answers. Manugi, read the question again. Right. What is the answer, my dear students? Which of the following is complementary event in meiosis? It must occur during meiosis. What's the answer? Answer is the first one. Why? Pairing of homologous chromosomes. Pairing. Because meiosis is a process of separation of homologous chromosomes. So therefore, pairing is a must. Right? Now, if you say crossing over, that's not a must. Right? And then formation of chiasmata is not a must. Formation of 4 identical daughter cells are not going to be identical in meiosis. Wrong. Duplication of centrosomes, no, you can't take that either. Answer is pairing, because this is a process of pairing and segregating of the pairs. All right. So that's the mechanism. The pairing makes meiosis different from mitosis. Mitosis, you don't see pairing. Meiosis, you see pairing occur. Right, then moving to next page, my dear students, moving to next page. Shall we move to next page? Right, question number 11. Question number 11. Which of the following is incorrect regarding proteins? Incorrect regarding proteins. Can you see the question? Oops. Which of the following is incorrect regarding proteins? Incorrect one regarding proteins. Myoglobin consists of several polypeptide chains. Hemoglobin is tertiary protein. Disulfide bonds present in tertiary structure. Keratin has alpha helical structure. Spider, the spider, please correct that. Spider, not cider. Spider silk has beta pleated structure. Spider silk has beta pleated structure. What is your answer? Which of the following is incorrect regarding proteins? What is your answer? Myoglobin consists of several polypeptide chains? No. Wrong. No. First one, myoglobin. Hemoglobin is consisting of, hemoglobin consists of 4 chains. Myoglobins consist of several polypeptide chains. Wrong. Myoglobin has only one chain. Hemoglobin is a tertiary protein. It is tertiary and quaternary. The tertiary level is there. Quaternary level present too. Hemoglobin anyway. Right. Myoglobin consists of several polypeptide chain. This is wrong. Several is wrong. Right. I'll take that second one as the doubtful statement because it has a tertiary as well as quaternary structure. Hemoglobin disulfide bonds present in tertiary structure. That is correct. Keratin has alpha helical structure. Keratin has alpha helix. Yes. And then spider. Spider silk has beta pleated sheet. Correct. So we have the best answer. I'm taking first one as the best. First one as the best. All right. Right. Moving to next question, people. Question number 12. Now, second is an answer, right? But the best answer is first. Because hemoglobin has a tertiary structure as well as a quaternary level structure. So the final structure of hemoglobin is quaternary. Right? Now, does hemoglobin has a primary structure? Does it has a primary structure, hemoglobin? Yes or no? Now answer me. Does hemoglobin has a primary structure? Yes. Does it have a secondary structure? Yes. Hemoglobin has a tertiary structure? Yes. Quaternary structure? Yes. Now, I agree. I do agree with you, right, that the second statement is doubtful. I agree with that, okay? Right? Now, it's better if they had given it as well. Hemoglobin has a tertiary structure. Then you are correct. Is a tertiary protein somewhat questionable. But then myoglobin consists of several polypeptide chain is wrong. Why? Myoglobin is a monomer. Hemoglobin is a tetramer. So, what is the answer for the 10th one? 10th one. First one. Question number 12. Question number 12. Which of the following structural, which of the following structure site does not contain RNA? Which of the following structure site does not contain RNA? Ribosome. Following structure site does not contain RNA. Ribosome, lysosome, nucleus, chloroplast, cytosol. Structure or site does not contain RNA. Ribosome has RNA. Lysosome, no. Lysosome, no. Nucleus has. Chloroplast has. Cytosol has. So answer is clear. Second one. Answer is second. Right. Question number 13. Not a feature unique to prokaryotic cellular organization. What is your answer? Can you tell me your answer, Lamai? Can you tell me your answer? What is the answer? Not a feature unique to prokaryotic cellular organization. Presence of 70S ribosomes in the cytoplasm. Oh, 70S ribosomes in the cytoplasm. Questionable, ne? This is doubtful. Why 70S ribosome in the cytoplasm? Directly in the cytoplasm is not present in the eukaryote, only present in prokaryote, but they are present in the mitochondria and chloroplast. Membraneless subcellular structure. Membraneless subcellular structures. Extracellular flagella. This is correct, ne? Extracellular flagella, correct. Circular DNA located in the cytoplasm, correct. Cell wall with peptidoglycan, not a feature unique to. This is wrong, ne? Right? This is wrong. So the best answer 5. Process of 70S ribosomes in the cytoplasm. That's an issue. Anyway, you have the answer, right? Fifth one is the answer, right? Unique of prokaryotic cell organism. Cell wall with peptidoglycan. We know some has, some are not having that character. 70S ribosomes in the cytoplasm. You can take it as unique. It's present in cytoplasm. Otherwise, right, 70S. So I'm taking fifth one as the answer. I think you do agree with me. Question number 14. Select the correct statement. Next one. Select the correct statement regarding nucleus. Correct statement regarding nucleus. Present in all eukaryotic cells. Present in all eukaryotic cells. Select the correct statement regarding nucleus. Present in all eukaryotic cells. Nucleus present in all eukaryotic cells. Wrong, neither. You know, people, some eukaryotic cells, they don't have nucleus. Red blood cell does not have a nucleus. Phloem, C-tube elements, they don't have nucleus. So having present in all eukaryotic cell is a wrong one, right? I can't agree with that. It stains darker due to lipid component included. Stain darker? No, we didn't say that. Ne? And then nucleolus, now it's stained darker not due to lipid but due to protein. Nucleolus is surrounded by thin single membrane. No, double membrane bounded. Wrong. All types of RNA required to the cell are produced here. All type of RNA produced inside the nucleus. mRNA, tRNA, rRNA are produced here in the nucleus. Not wrong, neither. And then people, only a single prominent nucleus is located in the cell. Nucleus is located in the cell. No, you know, people, sometimes the multi-nucleated condition present. Can you give me one example of a multinucleate cell? A cell with many nuclei. Can somebody tell me one example of a cell with so many nuclei? Can someone tell me? Skeletal muscle cell. Say it like that. Skeletal muscle cell. Okay, good. Skeletal muscle cell. You can take fungal cells too. Fungal cells, right? So some examples are there. Question number 15. Question number 15. Right. Question number 15. Which of the following is not acceptable regarding? Following is not acceptable. Which of the following is not acceptable? Following is not acceptable regarding organelle function combination. Organelle function combination not acceptable, right? Ribosomes protein synthesis acceptable. Smooth endoplasmic reticulum synthesis of vitamin D. Smooth endoplasmic reticulum is involving in synthesizing of lipids. So vitamin D, exactly vitamin D question. Rough endoplasmic reticulum synthesis of glycoprotein, RER, correct? And then Golgi complex synthesis of glycolipid, wrong. We didn't say that. And then mitochondria, photorespiration, yes. So we have the answer, right? We have the answer. Right, smooth endoplasmic reticulum, synthesis of vitamin D. We don't have a direct clue for that, but anyway, people, lipid synthesis involved. Right? It carries out lipid synthesis. So I think fourth one you can select as the answer. Right, moving people to next question. I'm taking question number 16. Lavai, 16th question. Which are the following incorrect regarding peroxisomes contain reducing enzymes? Peroxisomes, so what sort of enzymes they have? Peroxisomes, reducing enzymes, so oxidizing enzyme. So, oxidizing enzyme. So, having reducing enzyme is a wrong statement. More abundant in cells having high metabolic rate. More abundant in high metabolic rate, correct. Participate in metabolic paths more widely in plants. Carries out detoxification, and then present in both plant and animal cells. Correct. So we have the answer. I'm taking people first one as the answer. Question number 17. The pyrimidine bases common for both DNA, RNA. I'm taking the answer. You know it straight away answer, people. Cytosine, third one is the answer. Question number 17. Question number 18. Which of the following is not a function of plant vacuole? Not a function of plant vacuole. What is your answer? Right, Lamai, can you send me your answer, my dear students? My dear students, can you send me the answer for that question? What is the answer? You have marked fifth one. Question number 18, fifth. Really? Which of the following is not a function of plant vacuole? Maintain water balance of the cell, correct? Gives turgidity to the cell, correct? Produce colors in some plants from stored carotenoid pigments? No. 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 Is it? Carotenoids or anthocyanin? Carotenoids or anthocyanin? Vacuole stores anthocyanin. Where does this carotenoid stored, people? Carotenoid is stored in plastids. You learn the type of plastid. We said that chloroplast, chromoplast, and there was another one, leucoplast. So, no. Stores water and other substances such as sugars and ions. Yeah. Stores soluble substances needed for cellular activities. Yeah, correct. Yeah, why not? Why not? It can store some of the important sugars and all, right? Later on, they can take it back and then use in cellular processes. Not wrong. Right, moving people to question number 19. Question number 19. Question number 19. Which of the following gives brick red precipitate? Brick red precipitate when boiled with Benedict's reagent. Brick red precipitate when boiled with Benedict's reagent. When boiled with Benedict's reagent. Maltose, yes. Lactose, yes. Sucrose, no. Fructose, yes. Amylose, no. So we have the answer A, B, D. A, B, D. A, B, D. Question number 20. Which of the following is true regarding interface of eukaryotic cell cycle? Cell growth occur in G1 and G2 phase. Cell growth occur in G1 and G2 phase. Which of the following is true regarding interface of eukaryotic cell? Cell growth occur in G1 and G2 phase. Yeah, correct. Histone protein are synthesized. Histone protein are synthesized in S phase. Correct. Number of chromosomes in the cell become doubled at the end of the interphase. Not at the end, neither wrong. During the S phase of the interphase. Now, S phase is not at the end, wrong. And then people, DNA content of the cell increases only in the S phase. Correct. And then increasing of the number of the organelles happen only in the G1 phase. Increasing of the number of organelles occur in the G1 phase wrong. I think organelle number increase is mentioned people for both G1 and G2. I think you agree with that. So what is the answer, people? We can go for A. We can go for A, B, D as the answer once again. A, B, D. Got the answer? Right. Good. Right. Moving now, people, for the structured part, Lamai, shall we go to the structured part? Right. Moving to the structured part. Right. Structured questions. Structured questions. Structured questions. Right. First one, people, in which criteria living organisms show wide range of variation, size. Where did you learn it? Introduction, Balini. Size. What else you say? Shape. Shape. And then you can see, people, form, habitat. Habitat, right? Right. Second question, which property of water is significant in following stages? Which property of water is significant in following states? Striding of some insects on water surface. What did you write? Cohesion. Cohesion. And then, people, cooling of plant body by transpiration. You are going to take, people, ability to moderate temperature, right? Ability to moderate temperature. I'll write temp. For temperature, I'll just write it as temp. And then, people, upward movement of water through xylem against gravity. Cohesion. Cohesion. The next one, mention the two major forms of exhaust sugars according to the location of the carboxyl group. What do you call them, people? Aldoses / ketoses. Aldoses and ketoses. Next one, mention compound of elements of following biological molecules. Keratin, you can write carbon, hydrogen, oxygen, nitrogen, and sulfur. Next one, cholesterol, people, carbon, hydrogen, oxygen. That's all, right? Okay, then moving, people, to next part. State the structural state of protein represented by enzyme lysozyme. What is that, people? Tertiary. Tertiary structure. Tertiary structure. Next one, nominate the group of organism consisting prokaryotic organization, bacteria. And the other one, bacteria. Bacteria and archaebacteria. Then next one, people, state one physiological feature shown by some of the prokaryotes. What is that, people? Nitrogen fixation. Nitrogen fixation. Nitrogen fixation. State the subcellular structures common to both prokaryotic and eukaryotic organisms. Common to both prokaryotic and eukaryotic, ribosome. Ribosomes. Name one chemical component found only in animal plasma membrane. Chemical component found only in animal plasma membrane. What did you write for that? Lamai. What did you write? I'm taking the answer as this one. Do you agree with me? Cholesterol. Very good. Next one, people, into which major group of organic molecules above mentioned? What is that? Lipid. Lipid. What is a major chemical compound of extracellular structure of the plant cell? Cellulose. Chemical component. What is a major chemical component? Cellulose. Ne? I was just thinking about whether to write carbohydrate for that. No, I think cellulose is fine, right? Question number 5, people state one chemical compound found only in the secondary cell wall of plants. What do you write there? Lignin. Lignin. All right, and then moving to next page, students. Next page, right here. Next page, right? Okay, right. Next one, people. What is cell cycle? What do you call cell cycle? Events that occur from the end of one cell division to the next cell division. How do you say that? Beginning of one cell division to the end of the next cell division. Um, yeah, the sequence of events that occur in the life of a cell from end of one division to the end of next division. Right? Occur events occur in the life of a cell from end of one division end of one division to the end of next division. To the end of next division. To the end of next division. Fine. Next one will, what is the overall result of components of the cell cycle of an eukaryotic cell? Formation of new cells. Formation of new cells. And next one, what is the overall result of completion of the cell cycle by an eukaryotic cell? You know, new cells are going to form. Mention the role of the following structures. Right, they have given people some structures here. One is kinetoko. What do you know kinetoko, people? Oh, the sequence of events that take place in a life of a cell. Yeah, correct. Mention the role of following structures kinetoko. We know people microtubules are attached to the centromere, right? Attaching microtubules to the centromere. Special protein for microtubule attachment. Special protein for microtubule attachment to centromere. Attachment to centromere. Next one, non-kinetochore microtubules. Non-kinetochore microtubules elongate the cell at anapase. Elongate the cell at anapase. Next one, synaptonymal complex. Hold two homologous tightly. Hold two homologous tightly. Hold two homologous tightly. Synaptonymal complex. This is for the crossing over exchange. Right, next one, people. Draw a cell with 3 pairs of chromosomes at metaphase. Now, this is critical question. I know that most of you have not answered this part, right? How many of you have answered this question, people? This part, draw a cell with 3 pairs of chromosomes. Right? How many of you have? Me. Black. Black says me. Anyone else? right? Check this one. This is how I draw it. Uh, metaphase of mitosis and metaphase of meiosis. So I'll take mitosis first. 3 pairs scale again. How many chromosomes should be there? 6 chromosomes. 1, 2, 3, 4, 5, 6. This is how you have to draw them, right? Like this. Did you draw it like this? Maybe the rest of the part you can complete. I'm just showing the basic thing. Draw a cell with 3 pairs of chromosomes and the metaphase of mitosis. Uh, it doesn't say that whether it is a plant cell or animal cell, so you can just forget about that. And then people in meiosis. How to draw in meiosis? Meiosis. Draw it like this. Show them like this. Alright, did you draw it like that? I'm happy with this answer. I'm happy with this answer. Right, next one, people. What are galls? Galls. What are galls? Right, what are galls will we learn? Galls are some of bumps formed due to uncontrolled. So do we need to consider? So do we need to consider? Can you show the diagram again? No, you show it like this. Now you have to complete the rest of the things, right? The spindle fibers, the other one, you draw it properly. I'm just trying to show you people. Here in mitosis, all 6 chromosomes, even if there are pairs available, they align like this, right? Then here the homologous pairing occur. If you use a color code or something, right? You follow the resource book way, but these are the two main ways. Here they are arranged like this in the metaphase of meiosis. Mitosis, there is no pairing, right? Same, no pairing. Right, next one, answer for C part. Right, question part 5. What are goals? What are you going to write, people? They are bumps and growths of bumps and growths that develop on different parts of the plant after induced by unique organisms, right? Galls are bumps and growths that develop on different parts of the plant after being invaded by some unique organisms. After being invaded by unique organisms. What do you call them? Gall causes, isn't it? After being invaded by unique organisms. You write that. What are they? They are the bumps and growth. Bumps and growths that develop on different develop different plant parts of plants parts of plants after invaded by unique organisms. Right, next one, people, what is metastasis? You see that spread of cancer cells. Spread of cancer cells to locations distance from their original site. To locations distant to their original site. Original site, the site of the beginning, original site. Then, people, I have given you an essay question to write regarding briefly describe result of rapid uncontrolled mitosis. Rapid uncontrolled mitosis. Right now, you learn, people. So, rapid uncontrolled mitosis. Here talk about, people, the formation of galls, and here talk about the what do you call it, formation of tumor, cancer, and gall. So there is a separate theory part in your resource book, right? There is a separate theory part in your resource book, right? And then you will write the whole paragraph, right? So these points should be there. Briefly describe the result of rapid uncontrolled mitosis. Rapid uncontrolled mitosis. I didn't prepare a market scheme, people. I didn't have time to go with the scheme. But I think you can write based on the points given here. Result of rapid uncontrolled cell division. So because there are some points here, but then you can't write the whole paragraph. You can see that the cell division is controlled by some of the external and the internal factors, right, by chemical and physical. And then sometimes some of the body cells are not going to respond to these the internal and external signals, and they start dividing by their own without the affected without depending on the growth factors produced by the organism. They produce um, some of their own growth substances and then divide by their own. But now that's not the question. Briefly describe the result. So you have to describe the result. What are the result, people? The tumors. So you have to describe the formation of the benign tumor, and then sometimes the growing of these benign tumors are now pressing the nearby tissues and then damaging. And then the benign tumors, some of these cells can spread from the original site to the other locations. Then it transforms into what? Into a malignant tumor, and the process is metastasis. So that part you have to write. Plus, people, you must write regarding the formation of galls in plants, including the yeah, so it forms the galls in plants, right? So give a small description on that too. I think you can write it. I'll send you a market scheme on that to the group. And then that's all for the day, people. And thank you very much for joining tonight. Then I'll meet you all again in the normal weekend class, right? So see you all. Ibon and bye bye.